Yes.
Okay. Section 13.1 is on page 360, and it talks about, this section is about how we use our knowledge of genetics to make medications, improve, cure diseases, improve our lifestyles. And we can do this through genetic engineering. It's also something that you can do and make a lot of money. So I'm going to show you some of the basic techniques that they use here in the next couple of days. One thing that humans have done for thousands of years is called selective breeding. The process by which desired traits of certain plants and animals are selected and passed on to their future generations. All dog breeds have been selectively bred over thousands of years. I don't know if you know about the history of the dog, but the species of, or the dogs, which is Canis familiaris is the name of the species. Dogs have only been around for about 10,000 years. They were originally bred from wolves. The dog species came from wolves. Wolves that were a little more tame, and that would follow around humans and beg for food and eat some of our scraps. And we learned to breed these dogs, and we chose traits that we were interested in. The Saint Bernard is a dog that has a really good sense of smell. Huskies can run for a real long time. They can make good sled dogs. They don't get tired very easily. German Shepherds are real good at listening to commands. And they're smart, and they can be trained very easily. So different dogs are bred for different purposes. Smaller dogs like Chihuahuas and things like that were bred to be able to chase rats into their holes and things like that.
Dachshunds for ferrets?
I don't know, but perhaps. Yes?
I was going to say, you know how they have those foxes that whenever they're in snow, their fur's like bright white?
Yeah.
But then when they go to the desert, like the temperatures are hotter, which gets fur. But why doesn't that happen with huskies? Whenever, like, usually their fur always stays one color.
I don't know.
In your eye. I'm just kidding.
So anyway, what selective breeding is taking a trait you're interested in and breeding that trait so that it becomes part of the population. Horses, this was done to. They took the biggest, fastest horse and mated with the biggest, fastest horse they found and got kids, and some of those kids were even bigger and faster than their parents. And they've been doing that for thousands of years, and now you have the thoroughbred, which is a big, fast racing horse, twice the size of wild horses. And that's all done through selective breeding. Yes?
Could you, this is like a serious question, if you bred like a Saint Bernard with a Husky, would you have like a dog that could smell really well and run really fast?
Yeah, that's called a hybrid. And the book talks about that. Yeah. Hybridization is the next chapter, and if you breed two traits from the same species, trying to get both of those traits in the offspring, that's called hybridization.
Cool.
And you often see that done, especially with plants. Say that there's a type of tomato that's disease resistant, and there's a different type of tomato that doesn't require much water. Scientists will take those two types of tomatoes and cross them to try and get a tomato that's both disease resistant and doesn't need much water. Now it's kind of difficult, sometimes it doesn't work. Sometimes you cross the two and you get something that's not very good at either thing. But sometimes it does work. So yeah, scientists and farmers and people interested in the field will do this with all sorts of things. One problem that scientists run into when they are trying to do this selective breeding is called inbreeding. You ever heard of inbreeding?
Yes.
Inbreeding is when you take two closely related individuals and mate them.
Is that like incest?
It's kind of like incest, yeah. The problem with it is if you take two closely related individuals and mate them, say there's just a family of dogs that's real easily trained, and you want to make more of those dogs, so you take this German Shepherd that's very easily trained and try and mate him with one of his cousins that's also very easy to train. Well, sometimes you might, by breeding those closely related individuals, you might get some recessive alleles together that were present in an ancestor, and they're also present in the kids, but those recessive alleles could kill the individual or cause problems that you don't want. So that's a problem with inbreeding, getting dangerous recessive alleles together. And that's why it's normally not done. Humans don't like to try it because that's you could end up having a death or a problem. Yeah?
Why is it harder to come by thoroughbred dogs?
Why is it harder to come by? Because they have to make sure, you have to have a pedigree which makes sure that they are only mating with the right types, that there's no other blood from any other organisms in there. And you got to really keep your dogs corralled and that kind of thing and make sure they aren't mating with anyone else to make sure you know what the kids are. And so there are just, families that do this with their dogs and have the pedigrees and make sure that happens. Just a dog that you find out there on the street you can't be sure about. Yeah?
What are the chances of inbreeding a dog? What are the chances that those alleles will hit?
I think it depends upon the species and how closely related they are. If they're brother and sister, that's high chance. Yeah, high chance of problems.
Yeah, why some German Shepherds or some dogs have hip problems?
Exactly.
What problem?
The hip problems of
Yeah,
that comes from inbreeding. Yeah.
German Shepherds inbreeded a lot.
Yeah.
Can dogs be retarded?
Yes.
How do you find that out?
I don't know. It's not as easy to spot as in humans. That's a good question. I don't know. But yeah, they can have similar problems that the humans have. Yeah.
All dogs colorblind?
Yes. To some extent.
So it's not completely colorblind blue?
I'm not exactly sure, but yeah, they can't make out colors very well. They're kind of like deer. You can wear an orange vest when you're hunting because the deer can't see orange. I don't think dogs can either.
Well, is it true that some people are trying to mix the breeds of dogs?
Yeah, like different
Like
breeds.
a bulldog and a shih tzu?
Yeah, that's hybridization. That's it. If you like the bulldog's traits and the shih tzu's traits, and you made the two, you're doing hybrid. It's called hybridization. Yeah.
I don't know, but for dogs, people starting clone stuff. What would happen if you cloned a dog and then mated it with that dog?
I'll show you what cloning is. I don't know.
Would that be inbred? Or
Yeah, that would be very inbred.
Yeah.
Mainly
That would be
inbred.
itself.
Mating with itself. Yeah. That's as inbred as you can get.
Yeah, that would be impossible.
I talked about test cross already. This is something Mendel did with his peas when he didn't know one of the traits, and he did a test cross. Do you remember that?
Yes.
A cross with a homozygous recessive, and that will tell you what the unknown allele is. I'm not going to re-explain it. If you don't remember it, just reread about it. I think you can take it. A test cross, yeah, you can do a Punnett square to see what the results will be. Now, this is a glowing
Caterpillar.
mosquito larva. Is it caterpillar?
Looks like a caterpillar.
No, mosquito larva.
It says mosquito.
You can see the glowing mosquito larva in your book on the bottom of 363.
That's different.
Even cooler, I think, is the glowing mouse embryo.
Ooh. I like glowing mouse.
But is that as cool as the glowing fish or the glowing tobacco plant? They are all extremely cool. Now, how do you get a glowing fish, a glowing tobacco plant, a glowing mouse embryo, and a glowing mosquito larva?
Genetic engineering.
And by the way, you can buy these. You can order these right now off the internet. Look it up, glowing fish, yes. You can order glowing fish.
I can get a glowing rat.
They're not even that expensive. Listen. Here's how you do this. This is a form of genetic engineering. You take a gene out of an organism, like a jellyfish that glows in the dark, and you put it into another organism, like the fish here, or the tobacco plant. And so the fish will use the gene for glowing that you got from the jellyfish, and it will glow. And it just does it normally. It doesn't hurt the thing. It glows. Actually, with the fish, you have to feed them special food, and only then do they glow. But it's still pretty cool. Yes, Graham?
My cousin's friend is a glowing frog.
Glowing frog.
Like, I've seen it. It's weird.
Sweet. Now let me show you how they do this. Now, yes.
The things you showed us, they're like kind of small. Like I've seen a picture of like a glowing cat.
Are you
Is it harder
serious?
to do in larger organisms?
I don't think that's a real. I don't think that was real. I've never heard of it having been done in a cat. Yes.
You make a human glow.
You could theoretically.
No, man, you're just.
How's that try?
Well, the problem is when you try to do this, usually the organism doesn't grow and become glowing. It takes a lot of tries before they get it right. And that's fine to do with tobacco plants and fish and mosquito embryos. When you start trying it with humans, and humans are dying because of the procedure, then you get in a little ethical problem. Yes?
That's a plant. Can other plants grow or just the tobacco plant?
They could do it in any plant.
Really?
Yes.
Well, how do they do it?
I'm going to show you. That's what I'm going to do right
Why
now.
don't they try it?
Is this our other essay?
Death row.
Yes, there is an essay about this kind of thing. So, glowing is just a way of showing that the technique works. They don't always use it to make things glow. For instance, if they find a plant in the rainforest that makes a protein that causes it not to be eaten by pests, they think, hey, wouldn't it be nice if our corn plants had that protein?
Yeah.
Where insects won't eat the corn plants. Then you don't have to spray them with pesticides.
That's right.
So if you can take the gene out of the rainforest plant, put it into corn plants, then all of a sudden you have corn plants that don't need to be sprayed with pesticides. Think of all the corn farmers in the world. They're going to buy their corn from your company that produces this. And you can get rich that way. You see? They're trying this with all sorts of things, moving genes from one place to the other. There's a case where it's been done in humans. They took the insulin gene from a human chromosome, the gene that produces insulin. Do you know what insulin is?
Yeah.
Insulin is stuff that your pancreas squirts out right after you eat. After you eat, your pancreas squirts out some insulin, and that makes the amount of sugar in your blood go down. See, right after you eat, your blood sugar is real high. But people who are diabetic can't make insulin. So, they get high blood sugar after they eat, and then they have to take injections of insulin, which works. Works fine. But where do they get the insulin from? They have to buy it. And the places that get insulin usually get it from pigs and cattle. Pigs and cattle they can come by because we slaughter them all the time for food. And we can use their blood and strain out of their blood insulin. However, some people are allergic to pig and cattle insulin. And they can't take the insulin. And so they just have problems and end up dying at a very young age. Also, it's kind of expensive to get pig and cattle insulin. So what scientists did was they took the gene for human insulin and put it in a bacterium. You know what a bacterium is?
I know.
A bacterium, let me skip forward a couple slides. A bacterium is a very tiny organism. It's microscopic. Pay attention here. This is your essay. A bacterium is a tiny microscopic organism that has DNA, a looped chromosome, that's its DNA. And it also has a special tiny loop of DNA called a plasmid. In that plasmid there, they are able to remove the plasmid from the bacterium. And I have that plasmid on the board up here. This plasmid here is right here. Can y'all see?
Yeah, verse.
Is it like an O or like a D?
It's a circle. It's a circular loop of DNA. You see how I have a little bit of it shown here? The G's and C's and A's and T's?
Yeah. Cool.
And they remove one of the chromosomes from a human cell. That's one of the chromosomes. So they've got a plasmid here and a human chromosome here. The next thing they do is they take a special protein called a restriction enzyme. And your book talks about restriction enzymes on the bottom of page 364. A restriction enzyme, this one I'm going to talk about is called EcoR1. A restriction enzyme looks at the DNA and reads the DNA and cuts the DNA like scissors.
In half?
Cuts it, I'll show you how it cuts it. And it cuts it wherever it sees the sequence GAA-TTC.
I see it.
Now GAA-TTC is a palindrome.
Yeah.
Do you know what a palindrome is?
Yeah.
Yes, what is it?
It's so you can spell backwards, right?
It's something that you read forward and backwards
Race
the
car.
same. Like mom.
Or race car.
Wow.
Or race car.
Wow.
That's good. That's good.
Yeah.
Race car, race car. Mom, mom.
Wow.
Wow. Wow.
Dad.
A.
Dad.
That's my joke.
Yeah.
I always tell. Didn't get much of a laugh this
I don't get it.
I love it.
How
though.
is GAA-TTC a palindrome? Well, GAA-TTC on the other side is GAA-TTC. So this spells kind of the same thing forward and backwards. It's a palindrome of sorts. And EcoR1 will cut the DNA wherever it sees that. So they take the plasmid out, and they put EcoR1 restriction enzyme onto it, and it reads the DNA, GAA-TTC, and it makes a cut right there when it reads that. And the other one reads the DNA in the opposite direction because, remember, the strands are anti-parallel. And it reads GAA-TTC in the opposite direction and makes a cut right there. And so the way this thing cuts it, it's going to come apart.
Whoa. What, do you just cut that?
What a sick.
Do you see?
Is it feeling okay?
And they're going to open up this plasmid by cutting it apart. So now the plasmid has been cut open. They then take the human DNA and they put the restriction enzymes on that too. And the restriction enzymes will read the human DNA, and wherever they see GAA-TTC,
Oh, I see it.
they're going to cut. So let's read down GAA-TTC. There's one. Cut. Snip.
Wait, why are you cutting right there?
Because that's just the way it does. It always cuts between the G and the
Does
A.
all DNA have GAA-TTC?
Nope.
No.
Doesn't.
Only some.
Only some. But it happens to be located right here, GAA-TTC, right above the insulin gene. And in another location, GAA-TTC, click, click, right below the insulin gene. And so what scientists have done is they've cut a piece of human DNA out from the human chromosome. This is the code. All these letters right here, that's the code for making insulin.
No.
Your pancreas cells read that code and are able to make the protein insulin.
So they can make it once I read that.
So if this sequence is read, and by the way, the sequence is actually about 1,000 letters long.
Oh.
It's got GAA-TTC on each end, and in between is about 1,000 letters that I don't have up here because that would be too big. Y'all understand? So imagine that this piece is real long. Yes, Grant?
2 questions. 1, that pink thing, the GAA-TTC, is that like DNA?
Yes, this is DNA.
DNA is DNA.
Okay. And
This is the DNA, human DNA, and this is the bacteria's DNA.
Right. And since you're cutting out like the insulin gene,
Yes.
is that hurting the human being? Like,
No, because they just removed one of the human cells,
Uh-huh.
could have been a skin cell, and they pulled the chromosome out of that cell, so it doesn't hurt you. You got plenty of cells.
Okay.
Good questions. Good job.
You have trillions of cells.
Yes, you have 80 trillion. You can spare a few.
Just a few though.
Okay.
Like my brain cells.
So now the human insulin instructions will be moved over here, and if you will notice, There is a jagged end here. The way this restriction enzyme cuts, it leaves a jagged end. This TTAA is called a sticky end. And it will stick to an AATT over here. And these things fit together like so.
Love. You guys like click, bro.
Click.
You gotta redo it. Gotta redo the whole thing.
Start possibly.
There's a special enzyme that I don't know if we learned yet. It's called DNA ligase that comes in and actually takes this cut piece. See, I drew little hands on it, and it pulls them together. Click.
There you go.
And it pulls them on this side together. Click.
There you go.
This enzyme is called DNA ligase, and it's needed to do this.
Does it just hop over it?
Not too.
Click. This is a three-dimensional structure, so it can go around it.
Okay, just check. Alright,
What were you saying?
Okay. Now we have.
Now zoom in on the projector so we can actually read it.
Oh, Chris.
So listen up. Let me show you what we've done. We've taken the human DNA, cut out the insulin gene. There it is. We've taken the plasmid from the bacteria, cut it open, insert the insulin gene into the plasmid. Now we have a plasmid, a ring of DNA that has human DNA in it. It's got human DNA and bacteria DNA. We call that recombinant DNA. Recombinant DNA has DNA from two different species, the human and the bacterium. We're then going to take this and put it into a bacteria's body.
I got a question.
Yes.
It's gotta be pretty hard to find that bacterium. It's microscopic.
Well, you know what they do? That is a good question. They don't just take the plasmid out of one bacterium. They take the plasmid out of millions of bacteria. And they take millions of insulin genes. And they put them, they mix them all together in a test tube. And it just so happens when you mix them all together, new bacteria will suck in the plasmid into their bodies. Bacteria will pick up any floating DNA that they find laying around, and they'll pull it into their little bodies. Yes, Taylor?
How do they pull out like the plasmid and stuff?
Well, all they have to do is they give a chemical that destroys the bacteria,
Okay.
and the plasmid will just fall out.
And then destroy the plasma too?
No, it doesn't destroy the plasmid, it only destroys the bacterial cell wall.
That's awesome.
Good question, it's a special chemical. Y'all are asking great questions today. It's good. Okay, so we put the recombinant DNA, listen, we could put the recombinant DNA back into a bacterium, and now we have one bacterium with insulin instructions. And that bacterium will make some insulin. But one bacterium doesn't make very much insulin. But
Billions.
the bacteria will copy itself when it divides. So all you got to do is feed the bacterium, and it will divide, and you go from 1 to 2.
What does it eat?
Then from 2 to 4. 8, 16, 32, 64, 128, 256, 512, 1024, 2048, 4096, 8192, 16384, 32768, 65,536, 131,072.
Are you a robot?
Pretty soon, you'll have billions of bacteria, each producing a little bit of insulin. You grow them in these huge containers in a factory, and you suck the insulin out of the containers, and you bottle it, and you sell it to diabetics. And guess what? It's not cow insulin, it's not pig insulin, it's human insulin, so no one's allergic to it. You saved the lives of millions of people, and they're paying you for it. You're living on your own private island. You're a hero, and you're rich.
Got a hot white.
See?
Got a hot trophy jet.
Either that or you can speak golf.
Video footage.
Oh, you did.
Oh, wait.
Or it could be in the
I hit the wrong one also.
Scientists.
Hold on. Video footage.
Never.
Shouldn't have done that.
Watch.
It's good again.
Okay, this is the bacteria. Look, this is the bacteria, that's the plasmid. Watch.
Is that real?
Yes, it's really nice.
In a recombinant DNA technique using a plasmid vector,
Yes, sir.
foreign DNA is spliced into a bacterial plasmid. The recombined plasmid then carries the foreign DNA into a bacterial cell, where it is cloned when the cell reproduces. If the foreign DNA contains a gene for insulin production, each transgenic cell will make insulin.
Okay, that happened fast. Let me show you a little bit more slowly. There is the bacterium. In this case, they use E. coli. It's a common bacteria that's very well understood. And that's one of the plasmids. They pull that plasmid out.
Why are there two plasmids?
Bacteria have a number of plasmids. They pull that plasmid out. They cut it open.
Cleavage?
Cleavage with restriction enzymes. They put in the insulin gene that they had cut out from a human DNA. Then they put that recombinant DNA back into another bacteria. We call this cell a transgenic cell because it has DNA from a human in it. And then it multiplies like crazy if you feed it. And each one of those is producing insulin, that purple stuff that's coming out of it.
2, 4, 8, 16. See, there you go. That's you, bro.
Now this shows the process, the manufacturing process at the plant. How they do
Scientists
it.
begin with a cell from one organism. In one of the chromosomes is a gene they are interested in isolating. They put the cell in solution.
That's a
The
human
cell wall
cell.
ruptures. The chromosomes spill out. Molecular biologists then use restriction enzymes to cut the gene of interest out of the chromosome. The scientists can then transfer the gene to another organism.
That's the insulin gene.
One organism commonly used because it is well understood is a bacterium called Escherichia coli. In addition to having its own chromosomes, E. coli, like most other bacteria, have small loops of DNA called plasmids. Scientists can remove a plasmid, cut it apart, and then chemically view the selected gene into the plasmid. The plasmid can be put back into the bacterium, which will go on living its life, duplicating itself. But with one important difference. Each new bacterial cell now contains genetic information from 2 organisms. If scientists splice a human gene into the bacterial plasmid, the new generation of E. coli will be able to make a human protein. This basic technique of genetic engineering has many applications. At Eli Lilly and Company in Indianapolis, the first genetically engineered drug approved for human use is being produced. Here, scientists isolate the portion of the human genetic information that regulates blood sugar. They take the human genes, insert them into E. coli, and use that genetically engineered bacterium to produce a human protein, insulin. A technician is spreading the altered bacteria onto a medium. In order not to contaminate the sample, he's working at a lab station with an overhanging ventilated hood. Given time, the E. coli will reproduce, forming a number of individual colonies. The technician chooses a single colony, which will become the parent of an entire batch of insulin. To make enough human insulin for commercial purposes, Eli Lilly must encourage the bacteria to duplicate themselves many millions of times.
Sophie Drew watch video.
As the culture grows, it is transferred to larger and larger containers, ultimately reaching computer-controlled fermentation tanks.
The bacteria are living in those white tanks.
When production is complete, the insulin is chemically separated from the E. coli, purified, quality tested, made into a solution in huge vats, and then under sterile conditions, bottled for sale.
Put the insulin into these little bottles. costs about $80.
There is a growing demand worldwide for insulin to treat diabetes, which can be a fatal disease. Of the nearly 2 million diabetics in the United States who use insulin, most are treated with insulin derived from pigs and cattle. But some patients can't tolerate the animal hormone. For them, the genetically engineered human insulin is an advantage.
Imagine, that's a big money maker.
How do you get diabetes?
What's that?
How do you get diabetes?
Why can't they just put the bacteria?
Born with diabetes, it's a mutation.
Why can't they put the bacteria with the?
If they put the bacteria in someone, your white blood cells will kill it. That's an infection. You don't want bacteria inside. Yeah. No, wait for the bell to ring. You only got a minute. Let me tell you some of the other things they're doing. Listen. The strongest material made is spider webs. It's very strong, it's very light, and it's bendable. They're trying to make bacteria that can make spider web material. You take the gene for making spider webs, you put it in the bacteria spit out a bunch of spider web material. If you were to weave it into clothes or something, a shirt this thickness would be just as light, and it would be totally bulletproof. Think of the application that would have with soldiers or police.
Unless you're a
Wait, are they doing that for
Think of if
America?
you lined a tank with that material, how light it would be. This is the kind of thing they're working on right
Or
now.
cars.
That'd be the coolest.
Cars.
That'd be the coolest job.
Bro, is the United States doing cars for people living?
Why do you like bumper cars?
Anyone in genetics is doing it. One of the biggest leaders in genetics is Japan.
So that's not good. That's not good.
They're trying to do other things. They're moving genes around from plants, animals.
So they get